Hi, I'm Ken McGrath from Microba, and I want to run through a little bit more about the different kinds of tests that are available for looking at the gut microbiome, and just spend some time talking about how they differ. So to start with, gut microbiome testing and the gut microbiome itself has really undergone a pretty big change in how it's been perceived in the last 20 years. It wasn't that long ago when we thought that pretty much the only good bug is a dead bug and that all the microbes are pathogens and doing bad things. But really, it's quite apparent that's not the case. We now know that in us there's a community of organisms, many of them beneficial, helping out with our body and keeping us healthy and happy. That's not to say that there aren't bad ones too, but this community, and together it tends to drive our health and wellness. We now know that there are many different systems that are associated with the gut microbiome, and we talk about the gut-brain axis or the gut-kidney axis or the gut-liver axis, the gut-heart axis. All these are different systems which are influenced by the gut microbiome. And there's an overwhelming amount of evidence now to show that there is a connection between what the gut is doing and our overall health. So it's kind of obvious that we want to test this microbiome and really see what it might be doing for different patients to try and get a better understanding of if it could be their gut that's contributing to their overall wellness. So up until recently, one of the only ways we could actually test the gut microbiome was looking at the microbes through cell culture. And this involved taking a stool sample and planting it out on a bit of agar and growing it up and just seeing what grows. Now, it was the only test available up until the mid-80s, and so it was the best technology we had at the time. The limitation with it is that only a few microbes actually grow in culture, and around 5% of organisms are able to be cultured with standard techniques. Now, there are other techniques, you can get that up to around 30 or maybe 40%, and the number's increasing, but they're quite specialized culture techniques. And the standard CDSA test you might order really is looking at just a small subset of organisms. Still, with those, you can work out what is growing, and you can get decent sensitivity. Even as few as just one cell on culture can grow into a colony, and so you can start to characterize that. But you've got to keep in mind that with those culture-based tests, you're looking at a quite limited range of the microbiome. Now, another test that's available is something called a PCR-based test. These PCR tests are really quite focused, and what they're doing is looking for specific organisms present in the sample. The way they work is that people study the DNA of the organisms and they know what they're looking for, so they design a panel to PCR amplify specific regions of known organisms. It's important to realise you're not looking at the whole microbiome here. You're looking at a panel, and through a targeted assay, looking at if it's present or not. Now, that would be a multiplex PCR, so you might be looking at, say, 10, 15 or even 20 different organisms in a single test and seeing if you've got detection or not. You could also be looking at qPCR, quantitative PCR, which lets you look at the amount of the organism that's in the sample and gives you relative abundance information. But again, it's only looking at specific target organisms. You don't get a picture of the whole microbiome. At Microba, we often receive questions from practitioners about how our metagenomics sequencing is different to the 16S-based technologies. And so I really want to highlight these differences in more detail here. One way you can get a picture of the entire microbiome is by using something called 16S, ribosomal RNA sequencing. And so the 16S gene is a gene that's pretty common in all bacteria and other organisms as well, like archaea. And it's conserved, and what that means is that as these organisms have evolved and they have their evolutionary journey, the 16S gene tends not to change that much. In particular, most of the ribosomal RNA gene sequence is identical in certain regions. But there are parts of the ribosomal gene that are variable, and it's these variable regions we can look at to determine if it's a different organism or not. So the advent of 16S sequencing was actually quite a landmark in the study of microbes. It gave us a way to start to characterize the different organisms and use a fairly information-based approach to differentiate between the different species. The thing with 16S is that the gene itself is quite large. It's 1,500 base pairs long. But when people talk about 16S sequencing, they're typically only talking about a specific region of the 16S. One of the most popular ones is the V4 region, which is the variable region, the fourth one in the entire gene. And that region's only around 150 or 200 base pairs long. It's a tiny little region of the entire gene. And so from that region, you try and infer what the identity of the species might be. And unfortunately, that requires a bit of guesswork. You really can't accurately discriminate between different closely related species by looking at such a small part of the DNA. What this means is that typically you can only get genus level information from a test. You really aren't able to discriminate between the different species that might be present because that region is just too similar amongst those different species to tell the difference. The more advanced way and the more modern tool to use for profiling a microbiome is called metagenomics, and that's the technique that Microba uses. Now, metagenomic DNA sequencing is also called whole genome or shotgun DNA sequencing, and what it's doing, it's looking at the entire DNA genome. So not just looking at a small region of the 16S gene, but looking at all of the genes that make up all of the organisms in a sample. And that does two really important things. Firstly, it gives you very accurate species level information, so you know with a lot of precision which species are in each sample. It also gives you functional information, so it lets you know what those organisms are doing in the gut. For example, is this organism able to produce butyrate and hence helping gut health? Or could it be producing lipopolysaccharide or trimethylamine, things that are actually affecting the body negatively and hence having a detrimental impact on health? And so through that, you actually get a lot of functional information about how a person's gut microbiome might be contributing to their presentation in your clinic. The other thing about metagenomics is you can look at the organisms that aren't just bacteria or archaea. So you can look at the fungi and even the protozoa that might be present in a sample. So I just wanted to sort of explain that. And there was a study in the Science Journal back in 2016 that actually compared the kind of resolution that you're able to achieve using 16S-based sequencing versus metagenomics. And so if you're looking at 16S and looking at that tiny region, that small fragment of DNA, they determined that typically you can only work out what genus an organism is, and you're really only able to work out the species level for a handful of organisms. Now, for those specific organisms, you do get species resolution, and so you can hear people often say, oh, our technique using 16S does achieve species identification. So that's true for those few examples, but for the majority of organisms, you don't. You only get the genus level. However, with the metagenomic sequencing, you really get a complete picture and you hit species level IDs for everything in the sample. And the image on the screen now is just an indication of that. So what you're looking at here are different taxonomic levels starting from the center and moving out. And as you move to the outer ring, you're moving towards the species level identification. So you can see on the left-hand side, using 16S, only a few species actually hit that outer ring. So you're only able to identify those handful of species to the species level using 16S. On the right-hand side, you'll see the metagenomics approach, and pretty much every organism in that sample is identified to the species level. That outer ring is really complete. And this just shows you the difference in resolution between the two techniques. So the reason that this is important clinically is because it lets you differentiate between similar species that actually have different functions. So take, for example, Bacteroides. Now, there are many different species of Bacteroides, and two examples are Bacteroides uniformis and Bacteroides vulgaris. Bacteroides uniformis is actually a beneficial microbe and produces things like short-chain fatty acids, as well as anti-inflammatory compounds. Whereas Bacteroides vulgaris produces detrimental compounds like branched-chain amino acids. And if you were using 16S technology, you'd only be able to detect that Bacteroides exists. You wouldn't be able to discriminate between those two species. And so you wouldn't actually know if the person had the good form or the bad form, or both. Metagenomics gives you that discriminatory power. You can actually tell what the species is and what functions they're performing. And so clinically, that's far more useful because you can act on that with more specificity. Another strength of metagenomics is that it lets you identify unknown species that are in the sample. And yet, even though you know they're unknown, you can still detect the functions that they're performing in the gut. Microba actually looks at these novel species and we find that they're in everybody we test. And in many cases, they actually form a significant part of a person's total microbiome. Now, 16S-based tests wouldn't be able to report on those organisms. And so if you're not using metagenomics, you could be missing out on critical information that's really important to your patient's health. So recapping on that, metagenomics lets you identify who is there with a high degree of precision. You're able to look at the identity of all the species in the sample. And doing so really lets you know what's in that person's gut microbiome. And the level of detail really hasn't been around before, so it's a lot of information to take in. But within the tool that Microba provides is the chance to actually look at each of those species descriptions and learn more about them in the patient report. The other key thing that we provide is that function. So metagenomics lets you identify what those organisms are doing. We give you information on if the gut microbiome is producing these compounds that influence health. Things like trimethylamine, like hydrogen sulfide production, and like lipopolysaccharide. How much is being produced by your patient's gut microbiome through the genes that we look at that are the potential to produce those compounds? And so it is that proxy measurement. We're looking at the DNA to produce the compounds. It's not a measurement of the compounds itself. There is literature to show there is a strong correlation between the two. So we're confident that this is still a clinically relevant result to use in practice. The third thing that metagenomics can actually provide is that because we look at all the DNA, we know what different foods each of the organisms likes. And so these are the prebiotic fibers that feed them. By knowing, say, that Faecalibacterium can feed on pectin, we can actually say that if you're low in certain beneficial organisms, you can increase those prebiotic fibers and actually boost the level of those beneficial microbes in a person's gut. By doing so, you're shifting their microbiome towards a healthier state. And the food suggestions that Microba provides are actually tailored for each person's individual gut microbiome to nurture the good bacteria they have that are below beneficial levels. So that's a really powerful tool that we provide to focus in on those key microbes that are doing good things in your patient's gut. And you can use that information to really start to correct any dysbiosis and move their gut towards a healthier state. As always, if you have any questions, please get in touch with us at the healthcare team at healthcare@microba.com.